In this video, we will discuss about some of the challenges of the CAR T-cell therapy in the solid tumors. One of the major challenge is about this tumor heterogeneity. This is not only limited to the solid tumors, but in other tumors also. We have seen that tumor heterogeneity is a common thing. So, in order to deal with tumor heterogeneity, one of the approach is to use this CAR that has two single chain variable fragment, so that it can target two different antigens that are present or expressed on the tumors. Or instead of doing the single CAR with two single chain variable fragment, we can use bispecific CAR or transduce these T cells with the two different CARs, so that T cells will express uh, this CAR separately that can target different antigens, that is single chain variable fragment 1 or single chain variable fragment 2. And how they can decide which variable fragments they need to target, and this can be done using this by having the information about TSA or TAA. TSA is tumor specific antigen that are expressed only on the tumors, whereas tumor associated antigens that are expressed on the tumors and also on normal cells, but in the expressions in the case of tumors are higher. And the other approach that they can use is uh, bispecific T cells engager. That is, when it gets interacted, or the T cells will get active only when the CD3 and the single chain variable fragments get interacted with the tumor antigens, and this is known as BITE. And this is similar to, or somewhere related to, the actual T cells activations. The other way is this universal CAR, where what they will do is that they will switch the intercellular signaling functions. So, for examples, in this case, we can see that the single chain variable fragment, when it's interact with the tumors, or instead of so in this case, what will happen? The universal CAR, the they will add another single chain variable fragment that can interact with the receptors on that are expressed on the T cells, so that the single chain variable fragment, when it interacts with the tumors, it will trigger the intercellular signalings. Similarly, these things can also be done using the antibodies. That is, when the single chain variable fragment, in order to activate the T cells, single chain variable fragments are there, so we can use the antibody that can target those single chain variable fragment on the CAR T cells to activate the T cells, and thereby increase the activated T cells within the patients. The other approach or other challenge is this tumor infiltration. In order to deal with this, we can do the local delivery, and also we can use the CAR with FAP targeting, that is fibroblast associated proteins. And since the tumor microenvironment has more of this high fibroblast contains, and also this they are more cancer associated fibroblast. So targeting the CARs with the fibroblast associated proteins will reduce the tumor infiltrations capabilities. Or we can also, the CAR can also be used, or the oncolytic virus can also be used with this ECM modifying receptors, so that it can degrade the tumor microenvironment surrounding the tumors. So this is about the tumor infiltrations, and thereby increasing the tumor infiltration capabilities. And the third challenge is itself the tumor microenvironment. So in essence, it will cause the immunosuppressions, and it has a high immunosuppressions or immune inhibitors factors, such as TGF beta and this PDL1 receptors. So in order to deal with this, the one of the things that they can do is to block PD1 ligand that are expressed on the T cells through this anti-PD1 or PDL1 antibodies. Thereby, this PD1 receptors or PD1 ligand will not be able to interact with the PD1 receptors of the tumors. The approach is to inhibit the cytokine receptors or cytokine arming or cytokine receptors, uh, and at the same time they also express the CAR that can release the cytokines, thereby degrading the tumor microenvironment that can be seen in this figure. And the other things is about the change in the intercellular signalings. That is, when the PD1 or TGF-beta receptors interacts with its counterpart, that is TGF-beta or PD1 receptors, then the intercellular signalings will not be triggered. The other things is the to change the intercellular signaling. So instead of deactivations, when the PD1 or TGF-beta interacts with their counterpart, what it will do is it will activate the immune response. And these things, some of the examples are shown here, that by upregulating this c-Jun and downregulating this NR4A3 and TET2. So all these things are just an idea how they can improve the immune functions in the tumor microenvironment that is present or exist surrounding the tumors, thereby increasing the immune response. So I hope you understood some of the challenges and how it can be overcome, or the ideas that can be used to overcome these challenges of CAR T cell therapy in solid tumors. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.